what happened is that this cell culture procedure was actually a manufacturing process. And it was to manufacture polio vaccines. And John Enders, who developed it, he got the Nobel Prize for it. So then after he used it to help develop that vaccine, he used it to do some experiments with polio. Now, he didn't actually say in these experiments that these cytopathic effects prove that polio is in the sample at all. But after the Nobel Prize and the success commercially of the vaccine, and also, that the polio vaccine is in the mainstream attributed to be the greatest miracle of modern medicine, right? So that had quite a huge influence. And so virologists began adapting that procedure, which could manufacture a vaccine, also as the proof that that virus is existing or causing that disease in the first place. So it's a very efficient way to manufacture vaccines, that both the manufacturing process provides both the product and the proof that the product should be made in one step. And this is very analogous to the use of the PCR test in modern medicine, because that also is a manufacturing procedure. It's used to manufacture copies of genetic material. And it has now also been used as a way to provide proof of something, whereas it doesn't really provide that. It's never been validated, but there is the appearance and the misuse of this procedure in that way.
This is what I find crazy about it, is that from what I can understand from what you guys are saying, it's almost like them basically saying, we have this thing in a video game, and then we're testing something with the test for the thing in the video game, and it's coming up with the same result. You're like, but it's from a computer. Like, it doesn't have existence in reality. And like, yes, but this is what our tests are showing. And you're like, but how are you seeing that has any reality in like the world that we're living in? And it's so fascinating to go, these are the people that are, running these trillion-dollar companies and putting policies in place and all that kind of stuff. And when you guys kind of grill them on these things and say, why can't, if it doesn't exist outside of the cell, how does it transfer from, you know what I mean? It's like fascinating that you go, how is this still the current paradigm?
Right. Well, this is a fundamental problem in experimental biology, and probably in other experimental sciences as well, that for biology, it underwent this molecular biology revolution, and that began with the discovery of DNA and its supposed double helix structure, and then molecular tests started being developed and used. A lot of it is with antibodies, it's with genetics. PCR was a major contribution because that's why it was awarded a Nobel Prize. But what took place is that there was a shift in experiments from doing experiments with actual organisms like animals, bacteria or single-celled organisms, to doing experiments in vitro, meaning outside of the body, in the laboratory, in a petri dish, in a test tube. And so that's not, what happens in a test tube is not necessarily what happens in a living organism, in an ecosystem, right, in nature. But because of the power and convenience of all of this molecular biological lab procedures and tests and techniques, they could do experiments very easily. Like, they didn't have to wait a lifespan of an animal. They didn't have to, maintain the expense of keeping the animals to do experiments and such. So it was much more convenient and yielded more quicker results. And it was it took off like a trend. I mean, people thought, oh, this is how we're really going to understand, how organisms work. And what they lost in the way is that they never did confirmatory studies to say that what they found in vitro is actually what happens in an organism. And so it's lost its way, and it's producing all kinds of false information because it's an artificial simulated construct. So there's some crazy examples of this, by the way, which are done. So one is I was looking into the toxicity of graphene and graphene oxide because that was talked about so much. And it was almost impossible to find a study where they actually gave graphene to animals and then saw what happened to them. Instead, what they did is they put graphene in cell cultures and they looked for molecules that were supposedly a marker for this or a marker for that, right? These biomarkers. But are those things really, a marker? Like, have they really fully been validated? Does that really represent what would happen if graphene were in an actual organism? And they're drawing all kinds of conclusions based on these things instead of saying, hey, what happens in an actual animal, when that's the only accurate way to determine it, until there's some kind of model that really rings true. But that's never been shown to happen. They never compare the model to the thing. Now, with the variants of COVID, it's even more ridiculous. And I found out this from doing some investigative work on the CDC page where it's talking about variants and how they get characterized as being of concern or that kind of thing. And they never actually get the variant from nature. Instead, what they do is they, have like this computer video game where they say that the genetic sequence of the variant is different from the other COVIDs. But then they take that computer sequence, they use a RNA or a synthesizer in the laboratory, a machine, to make that different sequence, and then they put that in essentially a nanoparticle that is called a synthetic pseudovirus. And then they study that. Now, why the hell wouldn't they get the actual virus? Like, it's killing millions of people. There's plenty of it available. They could just get some of that and do the research. But instead, they go through this painstaking method to, construct a synthetic pseudovirus. Now, why would they do that if the real thing existed? Right? It doesn't make sense. And then they're drawing conclusions based on that, simulation. But, so they're basically making something that doesn't exist in nature, then studying it, and then using those results to say this is what happens in nature. Right? It's completely illogical. And, it's obviously wrong, but no one is looking at it this way. They're just saying, this is what I'm taught to do. This is where the funds are coming from. I can't find the real variant virus in nature. No one else can, or else they would be giving me a sample.